How many people do you think we can get out of bed, or at least in our time zone? What's your estimate? So, to my surprise, we have 13 followers, which is way above expectation. So, how many people do you think will log in?
I think we can be happy with 10 viewers.
10? I don't think we're gonna make 10, but we'll see. So okay, we'll do over under. I go under 10. I don't even know where to see the viewers, so I don't know if we're gonna win. So to everyone watching, welcome. Thanks for tuning in for the first ever Nutrition Tactics live stream, and this is going to be a journal club. So what is a journal club? It's very different from a lecture, for example. So we're going to discuss a paper, and we're not necessarily interested in what can we learn from this paper, but of course we will discuss that as well. But it's really about learning to read a paper. So in a lecture for 1 or 2 hours, the lecturer would tell you everything they know about beetroot juice. That's not the case here. We're going to go through this paper and then see how do we read this, how do we judge this, would we have done things different in the study design or in how to write it down. And it just really helps you to learn to understand scientific writing and become scientific literate, so how to read research. The target population, or at least the level of this journal club, would be a bachelor student. So journal clubs are performed in most university classes, and the level that we aim at is a bachelor student, so someone who has a little bit of background in science. Not every scientific term is completely new, but it's not like this will be some super advanced journal club where everyone watching is a PhD and we discuss every small detail. Now, however, we keep our eye out on chat. So if people ask a lot of, let's call it simple questions, then we'll address the simple questions and we'll make things a little bit more basic. If if chat comes up with a lot of very advanced questions, then we'll address advanced questions and up the level a little bit. So it is, to some sense, democracy. Whatever you guys are interested in, we'll adjust to that. So let's start with the journal club. Maybe one more thing that's important to say. Scientific papers are not always freely available. So in these journal clubs, we'll only use open access papers. So open access papers means that the authors of the paper have paid for the paper to be freely available. So whenever we share something here, it's because we're legally allowed to share it. We can't just randomly pick a paper and share it because it's simply illegal. That's a bit, well, I would want to say stupid, because I think science should be available for everyone. Isn't that, are you not doing science for the benefit of humanity? Why do you want to earn more profit on top of that? But that's unfortunately how things still are. So we will comply with the rules. So very briefly about me, my name is Johan Trommelen. I'm an assistant professor at Maastricht University. Our research group is interested in all aspects of muscle metabolism. That can be aging, that can be clinically compromised patients, but myself, I'm mostly interested in sports nutrition, and that will be the topic of this journal club. Milan, can you introduce yourself?
Yeah, so my name is Milan Betts. I'm a PhD student at the same lab where Jorn is working. I'm currently focused on the microcirculation of muscle and its role in muscle growth. So, for example, how important is blood flow to grow muscles? I'm currently focused on more of the older population. So we want to let's say, see if we can reduce muscle loss with aging. But I would also like to do a bit more stuff going to the more young and also a bit more trained guys. Um, so yeah, I've been helping Jorn with nutrition tactics for a couple of years now, and yeah, we basically work in the lab together and also outside of work. We still work together.
Yep. So if people are wondering how long will this take, no clue. I expect a minimum of an hour, maximum of 2 hours, but we'll see. Maybe we'll discuss a lot in the beginning, and then as things take long, that will go a little bit faster at the end. And then maybe next time we'll do this, we'll focus a little bit less on the beginning and more at other sections of a paper. Maybe a question for you, Milan. Where do I see chat? Okay, I figured it out. Perfect. Okay, so if let's see if we can find some comments from chat, and then we'll try to respond. So let's start. So we picked a paper, one because it's open access, two it's a paper that I'm one of the authors, simply because I'm familiar with the paper. So it's just a relatively easy paper for us to begin with. And it's also a paper we think a lot of people will find interesting. It's a relatively simple paper to read, so we wanted to start relatively simple. And I think after just watching one of these journal clubs, you'll have a pretty good idea of how we read a paper, and then you can apply the same trick to more complicated papers that we'll hopefully discuss in the future. So Milan, let's say you come across this paper online. What are the first things that you look at that will determine whether or not you would even read this paper, whether it's worth your time?
Well, one of the things is the title. So usually you obviously read the title to see if it kind of sparks your interest. Two other things are the journal, which journal is it published in, and of course also the authors. I think in this case, I really like the title mainly because it already shows the effect of the study. So it clearly says this intervention, so beetroot juice supplementation, improves this type of exercise performance in trained soccer players. So this is really, it already gives you all the information instead of a lot of other articles that sometimes say, that would say in this case, the effect of beetroot juice supplementation on this type of exercise performance in trained soccer players. Like that doesn't really tell you anything. So that is something I really like here.
So why is that a bad thing, Milan? Because to me it sounds like, if without spoiling the result of your study, it's like a little bit of a clickbait title, and it gets more people to actually look further. Isn't that a good thing? Why are you annoyed by it?
Well, I'm also annoyed by clickbait. No, I think in this case, also as an author, you want everyone to understand the message and you want to send the message across. So that is one point. I think another point is, as a reader, you just want to know if it's worth to actually spend time on to read. If there were already 5 other papers I read on beetroot juice supplementation in trained soccer players, and then this one comes out, then I'm like, okay, another paper that supports this, but then I might reconsider if I want to read the whole thing thoroughly, because if I've already read a bunch about it, then maybe I wouldn't spend too much time on it.
Yeah. And also, often when you write a paper, you need like a specific reference that a paper, for example, has shown a positive effect. And if I go to the reference list and I see like, oh yeah, this paper is definitely supporting whatever claim they are making in their introduction. About the title, we got a question: why is every letter in the title a capital? And that is simply determined by the journal. So every journal has their own rules about how the layout from the paper has to be. So for example, if I go down, you'll see here that everything is like one column, while I would say 95% of all papers have like a 2-column structure. That's not up to the authors, that's simply what the journal wants. And this journal just says we want a capital for each word. Why? I don't know. It is quite common. I think they believe it improves readability, but there's no real reason why it is good or wrong.
That's
So
actually a good question because I've never really thought about it because it is kind of weird. Yeah.
yeah. So yeah, you mentioned this yellow part. This yellow part. So I've highlighted things in different colors that I think are perhaps worthwhile addressing. So this yellow part indicates that yes, the title gives the result, and that's useful. So what's this blue part?
Um, Wait.
So the blue part. So the blue part. So I made these colors, so I'll address them. So this blue one just indicates what the intervention is. Now I don't think anyone would ever forget to write a title without their intervention. But what you do see quite often that especially students forget in a title is this pinkish part, which is the outcome of a study. So it's quite common for students to write the effect of beetroot juice supplementation in trained soccer players. But if that's your title, as a reader, you have no clue what the paper is about. Is it about exercise performance? Is it about cognitive performance? Is it about blood pressure? Is it about muscle growth? Can be about anything. So it helps to use like a color system like this to just have that mentally in your head and see, do I have the intervention? Do I have the outcome? Am I indicating what happens with the outcome? So is it a positive or a negative effect? And then often it's useful to specify the population as well. So these 4 elements, if you have these in your title, then usually it covers everything that you need to know in a title. Okay, so next step, authors. Is that something that you would look at when you come across this paper? So maybe it's important to say in medical science or biological science, PubMed is the system that's used the most. It's like the Google of scientific papers, and if you look for something, for example, you would enter beetroot juice and exercise performance, then you get a lot of hits, and then you see the title, the authors, and the journal. So those are really and the year of publication. So those are the only things that can give you some kind of information. Do I want to click this and actually read this paper? So we discussed the title. What about the authors? Does that matter, Milan?
Well, it shouldn't matter too much if you, in terms of quality of the study. If it's a good study, then it's a good study. Then the author shouldn't matter too much. But I think it does give somewhat of an indication if there are some big names on the author list. For example, here, Luc van Loon is a pretty big name in the field. Then you know beforehand that there would probably be quite some, let's say there will likely be a lot of facilities to do proper research, and probably there's more money involved to design a very good study. So I think it can give a bit of an indication, but it shouldn't matter too much in terms of the actual quality of the study.
Yeah, so I don't think you can judge a paper just on the authors. That's just an argument from authority. However, it is an indication, as you say, because in research it's quite simple. pretty sure there's plenty of people who are way smarter and have a better work ethic than me, but if they don't have the lab resources, so that's just money for the study and the equipment, they'll never be able to do a study that's comparable to some of the studies that we do in our lab. So the authors give some indication of, is this a lab that has good resources, and that just makes it more likely that the study is good. But you can never conclude, oh, Luc van Loon is on it, so then the study has to be good. You have to look at the study objectively. Some other authors on there, so I'm on there, I'm a co-author. That basically just means that I helped out on the study. The most interesting spots on an author list are the first and the last author. So the last author is the person who got the funding for a study. So again, research is just very expensive and it has to come from somewhere. This can be a university grant, it can be a government grant, it can be funding from companies, but the money has to come from somewhere. And whoever obtained the funding for the research, that is the last author. So he's really the person that made everything possible. Then the first author is usually a PhD student, and it's the person who basically led the study on the day-to-day basis. So they were in contact with all the subjects, did the bulk of the work, did often the analysis, did the experimental tests. So here that's Jean. Jean is a good friend of us. For anyone who's Dutch, Jean has an Instagram called @sportsciencepro, where he does a little bit of the same stuff that we do at Nutrition Tactics, but then in Dutch. So check that out if you're Dutch and interested in it. It's @sportsciencepro. What about the journal, Milan?
So I think nutrients is, let's say, a pretty mediocre journal in this field. So it's definitely not the highest, but also not the lowest. It doesn't mean that it tells you everything about the quality of the study. But it can say something about what to expect if you have like a really big study with a lot of really high-tech methods. Then usually it will end up in a higher impact journal. And if you have a more basic study, it will probably be more in the mediocre or maybe sometimes in a lower impact journal. I think it's also good to mention that impact factors are quite relative to the field. For example, sports nutrition, those are definitely not the highest impact journals that you would see out there. If you compare it to like cancer research or something, they obviously have way higher impacts just because a lot more people are doing research on cancer. So also if you publish, then more people will read it and will also cite that work. That's my take on it. I'm not sure if you have any comments on that, Jordan.
No, I agree. So based on nutrients, I think you would expect like, okay, it's a decent study. It's probably they probably didn't have some revolutionary findings and probably didn't have 600 subjects that would make the study super valuable or something. But it's also not in a very low journal where it probably got rejected at more higher impact journals, and then in the end they just are happy that they got rid of it. So this would suggest it's just a decent study. Again, every study you have to objectively look at the study. It's just an indicator. Okay, so we've looked at these things, and then you decide, let's have a look at the actual paper. Then you usually start with an abstract. So a lot of colors here again. So you're reading this abstract. What are you looking for, Milan? So let's start with what is an abstract, and then do you think this is a decent abstract or can be improved?
Before we go into this, maybe we still have a question on the author list, which is, I think, a pretty good question in the chat. So someone is asking, is doing an internship and is wondering, yeah, when are you able to get on the author list? And I think that there are, yeah, it's pretty difficult to say because there are no rules for this. If, let's say, if you are doing an internship and your goal is to be on the author list, then I would discuss it with your supervisor, and you tell them, okay, I'm a motivated student, I want to be on the author list, what can I do to make that happen? And usually you would need to, yeah, really get in some considerable amount of work in, yeah, to get on the author list. So it's not that you're just doing an internship and you're helping out on some test days and you are a co-author. It usually takes a bit more work, but yeah, like I said, I would just discuss it with your supervisor.
Yeah. So as an intern, you like I was like that. I thought I was being super useful, but really you're kind of just in the way. That's honestly how people often experience it. Um, I think, well, you can definitely earn a position on a paper as an intern. I think you did it, Milan, I did it. But you kind of need to overperform because you need to do a solid job because you want a degree. So you already need to do a solid job just to prove you deserve your degree. If you want to be on the author list, you basically have to overperform. That's what it comes down to. It also really depends on a study. If you're working on a project that has been running for 2 years and you do a 4 month internship, like you haven't made a significant contribution. Uh, let's say you have a 6 month internship and the entire study is finished in that 6 months and you kind of overperformed, then it's much more likely that you deserve a spot on that paper. So there are no hard rules. Just discuss it with your supervisor. Say, I'm willing to do whatever I can. Maybe they say, well, then you have to do a longer internship, or maybe you have to do some unique contribution where you figure out some advanced statistics, or maybe in the evening you do some lab analysis that other students never do. So it is possible, but again, it's really yeah, it needs to be discussed because there are no hard rules. It's the same thing really in science. So there's a lot of, like, your publication records is supposed to indicate how well you do as a researcher, but there's a lot of people who on social media act like they're an interesting research because look how much I publish, but it's very clear that they're just on a lot of papers, but they haven't really done something. They just kind of say like, oh, I'll be on your paper, you'll be on my paper, and then we're better off. So it's a little bit of the Wild West. What deserves a publication spot? To be really honest, so for example, I'm a co-author here. Just as an example, I was at every single test day here, and all these test days were performed outside of office hours, as we'll discuss later. This wasn't done at the university, and it wasn't done between 9 to 5. But even as a co-author who've done significant amounts of work, that's why I said the first spot and the last spot, like those really did the work. Getting that funding usually requires writing complicated grants and stuff like that, and the first author just simply does the most work. And honestly, everything in between is kind of like, I'm sure you did something, but there's plenty of cases where people didn't do something. When I look at a researcher, I really, in my head, is like, okay, how many first authors and how many last author publications does he have, and everything else is like, whatever. But people still like it because it looks good in numbers. But yeah, it's good that you're interested in being a co-author because it can really help you early on in your career, because then when you would apply somewhere, people are like, oh, okay, he got on a paper, must be a serious student. So I would definitely discuss it with your supervisors. Okay, so Milan, what's an abstract?
So an abstract is basically the summary of the paper. It's usually around like 250 words, but I think it also kind of depends on the journal. in this case, you already nicely formatted it on the usual format, which is like background or introduction, and then you say something about the methods, some of the primary results, and then just a conclusion, as you highlighted here in the different colors. So I think here in yellow, just a brief background, just enough to kind of understand why they are doing the study, or at least understand also their hypothesis. Hypothesis in pink, which is also quite clearly formulated here.
So just very, very quickly, hypothesis is just the authors think that something will do something. That's what a hypothesis is. So it's just basically you have a research aim, which is finding out whether or not, for example, beetroot juice does something, and then your hypothesis is yes, we think it does under these circumstances, or no, we don't think that it will work. And you can't just say abstract. We hypothesize this and this. You have to give some background. So in this case, in our paper, we said, well, there's some papers that suggest that nitrate ingestion, which is something in beetroot, can improve performance in recreational athletes. But we're kind of interested in seeing whether or not it also works in more trained soccer athletes. There's a lot of supplements that seem to work in low-level athletes, but then don't work or work less well in trained soccer players. So therefore we thought it was an important distinction, and then we thought, no, we actually think it might still work in trained soccer players. Then the green is relatively the boring part. It's the methods. We'll go into more detail in the methods later on, but basically what the study entailed is subjects did a performance test, and once they did it with beetroot juice, or at least beetroot juice with an active ingredient in it called nitrate, or they got beetroot juice in which this active ingredient nitrate was removed. Finally, you have some results. What I think is most important with the results is you don't just say, well, beetroot increased performance or there was a significant improvement, because that's kind of meaningless. You want to know how big was that effect. So here we specified was 3.4%. Because if you just say it's a significant increase, for example, then is it plus 0.1% or is it plus 400%? You have no clue. So in the results, you would like to see some specific numbers. And finally, a conclusion, which is just, one or two sentence with the main conclusion. Keywords. What's that, Milan?
So the keywords are basically additional words that you can give to the journal and to your publication for people to find your paper more easily. So for example, you often have keywords that are not already in the title or the abstract. So for example, in this case, soccer was used to describe the sport of soccer, but some people often also call it football. So it totally makes sense to also put football in the keywords. So if you are looking at football performance or whatever, beetroot juice, then you will still find this paper. I think also nitric oxide is an important metabolite that is not mentioned already in the title or the abstract. So I think that's also quite relevant to put here.
Yep. We got a question what this plus minus number means, and the simple version is it just indicates, let's call it variation. So here they saw that on average subjects performed 3.4% better when they had beetroot juice, but it's not like every single subject performed 3.4% better. Maybe one subject performed 5% better, other subject performed maybe 2% better, just as an example. So this indicates how much variation there was. So here, this number is relatively small. It's 1/3 of this, which indicates that it's not like one subject was 200% better and then someone else was only 3.4% better, because then this number would have to be much bigger. So just the general way to look at this, the smaller this is relative to this, the more consistent this number is for everyone, for all the subjects. Okay, so let's start with the actual paper. So first section is always an introduction. Sometimes it's called background, depends on the journal again. And the purpose of an introduction is like the long version of this yellow part, just to explain why this study is being performed. And at the end of an introduction, as a reader, you should come to the conclusion, yeah, I understand why the researchers want to do this study, and I understand why this is a meaningful study. So you just can't, as a researcher, say like, I want to investigate the effect of hugging a tree on muscle growth, because, well, good luck explaining to anyone why that makes any sense. So we'll have a look at whether or not this introduction does a good job, and also how an introduction is built up. But first I want to have a look at this first sentence, because it's quite intense, I would say. So, while nitrate and nitrite were previously considered inert byproducts of the nitric oxide NO metabolism, recent insights suggest that dietary nitrate can also serve as a precursor for nitric oxide through the nitrate, nitrite, nitric oxide pathway. Okay, so if that's your first sentence, you probably want to run away. It's very biochemical, very pathways. To be fair, this is probably the most complicated sentence in the whole paper. Everything else is relatively simple. But when you have something like this, my number one tip is don't try to read on, because if you get lost early, it's very difficult you only get more and more lost. So you should try to understand this one first. And if you do, then usually everything else is not that difficult either. So here, I would just Google this, try to see, can I find like a picture of this pathway? And then once you kind of understand the pathway, things will fall into place. So I have done that for you. Well, I've made a figure. So here you have an overview of nitric oxide metabolism. And what they were saying in that sentence was that nitric oxide is thought to have all kinds of performing enhancing effects. So this is ultimately what we want. And then we thought we could make nitric oxide from the amino acid L-arginine. Now, to convert this L-arginine into nitric oxide, you need oxygen and the enzyme nitric oxide synthase. So if you have these three things, then you can produce nitric oxide and you can get some performance enhancing effects. If you form nitric oxide, it will have its effects, and the result will be that it will form this metabolite, nitrite, NO2, and this one will get metabolized into nitrate, NO3. So these things were just seen as not useful byproducts of nitric oxide, when nitric oxide has already done its magic. So let's go back here for a second. So they were considered byproducts of nitric oxide. But then the second part says, more recently, it now seems that nitrate can also serve as a precursor, which is basically a building block for nitric oxide. So what they're saying is, it seems that it also goes the other way around. If you have a lot of nitrate, it can be converted into nitrite and then converted to nitric oxide. So now these are not unnecessary byproducts, but are actually useful building blocks. But what you can also see here is that the conditions for this route are different than for this route. Here you need oxygen to convert to nitric oxide, but here this conversion happens when there's hypoxia, which is just a fancy way of saying low oxygen. So when there's high oxygen, this happens, but when there's a low oxygen, this happens. And this also happens when there's a low pH, which is just means there's an acidic environment. Now, what is a condition where these things are active? This is this is active during intense exercise at the location of muscle. So during intensive exercise, the muscle won't have much oxygen and there will be a low pH because you're doing intense oxygen. So this effect, we didn't really see that this was happening, this going to here, because under normal conditions, this pathway isn't very active. Then they found out, oh, during these conditions, so during exercise, this will happen. So this pathway is way more important for performing enhancing effects because this is what's effective of activated during exercise. And then they're saying, that's interesting because we can get this nitrate, not just recycling from previous nitric oxide, but we can ingest foods that have nitrate, such as beetroot juice, that will form this. So theoretically, by having foods that provide nitrate, and you do intensive exercise, you should be able to produce nitric oxide, and that should, at least theoretically, have performance enhancing effects. So making a scheme like this, or at least finding a scheme like this, is really useful when you have a paper that talks about all these complicated pathways. It's very difficult to learn something new just from text. So try to find something visual that can really help. So that was the most complicated sentence from the whole paper. There's no other scary pathways or metabolites that they'll discuss. So again, the purpose of an introduction is to arrive at why this study is worthwhile doing. And an introduction, we always explain this, is supposed to have a funnel shape. So what's a funnel? It's like the reverse of a pyramid. So it starts very broad and then it gets more specific towards your research topic. So each paragraph should be more specific towards the exact research question. So we're gonna look whether that's the case, and then so that's the macro structure. So each paragraph should be more specific. Then within each paragraph, there's also more or less a rule that the last sentence of each paragraph kind of should summarize the paragraph or at least give the main point of that paragraph. And that means that you can ignore everything before the last sentence, and then the introduction should still kind of make sense. So we're going to look if those two things are true. Does this introduction start very broad and then becomes more specific to the study? And does the last sentence kind of give the main message already? So you don't need to read every sentence. So here they say, these elevations in plasma concentrations have been associated with improvements in exercise performance, suggesting ergogenic benefits from activation of the nitrate to NO pathway. So far, all we know is that if you have nitrate, it can activate the NO pathway, and that can hopefully increase performance. Okay, so now we're going to go to the next paragraph, and that should be more specific then. So you go here. As such, this work, in line with others, has established a functional benefit of dietary nitrate supplementation on exercise performance. So the question here is this more specific? And the answer is yes. So here they say this pathway should be able to increase performance. Then here it's more specific because now they mention how you're gonna impact that pathway. So dietary nitrate supplementation. So you went from broad to we have this pathway to how can you impact this pathway with supplementation. Okay. Next paragraph. Based on these findings in rodents, it could be suggested that the ergogenic effect, which means performance enhancing effect, of nitrate might be the most profound for activities that recruit type 2 muscle fibers. that is very high intensity exercise bouts of short duration. So type 2 muscle fibers are sometimes referred to fast muscle fibers that you use during high intensity exercise. So we went from very broad, we have this pathway, to how do we impact this pathway with dietary nitrate supplementation. And then now we become more specific because we say this performance enhancing effect might be most effective during a specific type of exercise. So we went from this pathway has a general exercise performance effect to a specific type of exercise performance. So again, we're more specific. Then here we see the last paragraph. As you can see, it's a very long paragraph. If I'm honest, I would have rather seen this split up into two paragraphs, but let's see what the message here is. So they start discussing about soccer. So soccer is, again, they're specifying high intensity exercise. Now we make it even more specific by mentioning a specific sport. Then they mention that the yo-yo intermittent recovery test level 1 test is an often used measurement to simulate soccer specific activities. So they're saying if you want to measure soccer, how do you do it? Well, you have to do this specific test that resembles soccer. Then here they say previous research has shown that beetroot, or at least nitrate, can improve running performance in recreational athletes. However, it's not as clear if beetroot can also be effective in more trained athletes. So here, extending on findings in recreational athletes, we hypothesize that it can also work in trained athletes. So it should always be clear from your introduction that what is the novelty of this study. I can't just do a study that has been done 3 times already. There has to be some kind of novelty. So here it is, there's some studies that suggest it works in recreational athletes. We want to see it specifically in high intensity exercise in subjects that are more trained. So this last paragraph, ideally I would have seen it split up in 2 paragraphs, because here the trick of, I'll just look at the last sentence isn't as clear, but still the overall structure is quite clear that up till here we discussed that high intensity exercise is interesting, then we specify it with soccer, and then we introduce our specific hypothesis. So it's very clear that there is a funnel shape. Just our other trick that the last sentence of a paragraph basically tells the whole story didn't really work because this was too long of a paragraph for that trick. Milan, do you have any thoughts to add? Because this is not your paper nor a topic that you're necessarily super interested in. When reading the introduction, were you completely lost? Kinda okay? Any thoughts?
No, I think I agree with that. It's like a really clear funnel shape. I think what I noticed with a lot of students, bachelor but also master students, is that they still kind of struggle with this. They often want to put as much information in the introduction as possible, which is definitely not the idea of an introduction. I usually say that you just want to give enough information that people understand what you're doing, that they understand your hypothesis, and that they will understand your study design. If you go into the introduction and people also start talking about a bunch of unrelated stuff, that is only distraction and not really adding anything to the story. So I think that is also something that is in this introduction. It's really well done, but it is something that you probably also see that quite a lot, Jorn, that I think it's a mistake that quite some students make.
Yeah, so for example, beetroot juice can potentially lower cardiovascular health risk which is super interesting, and there's lots of people who do studies on that, but it's just not that relevant to know here. So an introduction should just give enough information to understand why this specific study is relevant, and that beetroot has 6 potential other benefits that are unrelated. You don't mention those here. That's something you would do in a review article about all the effects of nitrate, for example. So yeah, that's a good point. So especially if you're a student, you don't impress someone by throwing as much information as possible in there. It's like the minimalism idea is, what are the crucial elements that need to be in there, and then remove everything else because it's just distracting. You don't want a reader to think, oh, this study is about health, or this study is about whatever, finance. No, it's about beetroot juice and exercise performance. Yeah, good addition. Next up, materials and methods. So why does this need to be in a paper? Because isn't this by far the most boring part of a paper? Because I can definitely tell you that about until halfway of my master's degree, I just basically skipped this.
I mean, I agree. It's not the most exciting part of the paper, but I think in the end it is quite crucial because you basically want to give enough information about the study or the study design that someone else could replicate the study. So someone else could read this paper, see how did you do this study and replicate it, and probably would see the same results. So that is why you often see a lot of maybe like boring details in the materials and methods, because yeah, you would need to know that if you want to replicate it. So for example, if you with a certain analysis, you want to give all the specifics like temperature and whatever, so that someone else could do the same.
Yeah. Okay, so very specific. They start with subjects. So what would you need to describe about subjects, for example?
Well, actually, funny story about this. The other day, some time ago, I was a tutor with the review scores, and a student included some papers, and he said with a couple ones that it was not specified whether it was male or female, and I was like, "There's no way it's not in the paper." So I double checked, and it was actually not in the paper. Like they didn't say male or female. Like you just had no clue. And yeah, like that is obviously something quite basic, but it is obviously important to mention really specifically what are the people that you are interested in. In this case, male soccer players, also in a specific league of Dutch football. So that also tells us something about how trained they are.
Yeah, so basically any in science we dislike, let's call it subjective statements. So if you say trained, then any researcher, like their first definition, the first thing they're going to say is, what's your definition of trained? And that can be, sometimes it can be some biological variable like your VO2 max or the amount of muscle mass you have. Or in case of a sports, like here, you say this is the level at which they're competing. So yeah, it methods, just as you say, it's very important that things are specified. You should just never have a question like, what is trained, or what is the sex of the subjects? Okay, so I've highlighted this, so I'll quickly address it. Papers, in my opinion, should be registered. So what that means is that before you perform the study, somewhere online in a database, you describe exactly what you're going to do. Basically, more or less the method section that you've written here could be in your registration. And why is this important? Because otherwise you can measure 20 things, and then let's say you do this study and we measure 20 performance outcomes, and then one performance outcomes we see a significant improvement, and 19 other things we see no effect or maybe even negative effects of Beetroot. And then we want, let's say we have shares in Beetroot juice, or we want Beetroot juice to work because then it's a more exciting paper, we get more references and more attention. Then we can just write the paper like, see, Beetroot juice improved this performance outcome, and then ignore all other 19. We just ignore it. Obviously, that's not ethical, but to prevent any of those problems, you should register your study, tell exactly what you're going to do, what you're going to measure, so that people afterwards can, when you publish a paper, they can see like, okay, indeed, they said they would measure these 20 things, and they report all 20 things. So it just prevents any potential shadiness from going on. So to make it quite frank, there's no reason to not register a paper. So if people are not registering something, best case scenario, my opinion, a little bit lazy. Worst case scenario, something might be going on. Now don't overvalue this. I'm definitely not saying everything is shady if it's not registered. I just don't understand why someone would not do it. So you just can Google this code and you find a website where once you've entered that information, you can see the date where the information of the study was entered. That should be way before the paper is published, of course. And that site, once you enter something, well, you can change something, but then the site will always indicate, well, they changed this on this date. So you can always go back to what they originally registered, and that basically should be how the study was performed. Sometimes you want to, or you have to change something because there's a pandemic and you just can't do the study the way you wanted to do it, but then it should be registered, explained why. I mean, science is only as good as we can trust things, and everything that prevent questionable practice, I think we should simply do. Okay, then study this.
We have a quick question in chat. I think it's a pretty good question. So, question is whether you would specify in the title that it's about male soccer players, because I don't think it's in the title.
Nope. No, we say in trained soccer players. So the title is basically the briefest summary possible of your paper. And of course, any kind of summary is leaving out information. So your title doesn't give the same amount of information as your abstract. Your abstract doesn't give as much information as your paper. And your paper doesn't give 10% of the information that's in the brains of the people who did everything. Like any reporting, you lose some information. So you just have to make choices what should and shouldn't be in there. I have little reasons to assume that the effect of beetroot juice on high intensity exercise is different in males versus female players. And therefore I don't think it's super important to specify the sex in the title, because I think the title is true. I think it's true for trained male soccer players and for trained female soccer players. However, if there was research that shows that the effect of beetroot juice is different in males or females, then it would be a crucial component. So it really depends on the topic which details should or should not be in the title. If it's not in the title, it of course still should be to be found in somewhere in the paper, and luckily it is. Male soccer players. Okay, study design. So this is so typical research. Just throw a bunch of complicated words in a way too long sentence. So can you briefly explain what all these things are, Milan, and then whether or not it's good or kind of dumb?
Sure. So we have a double-blind study, which is in this case really important because it means that both the research participant but also the researcher, that they both don't know what they are getting, whether they are getting the beetroot juice or the placebo. So in terms of how good this is or how important, it's very important. It is the yeah it can already influence the results if someone knows what they are getting, if they are getting the beetroot juice with nitrate or the beetroot juice without nitrate. You also have randomized, which basically means that they get the treatments in a totally random order, which is also something that is quite important in research.
But so why is that important? Because, well, especially with something like this, like a performance study, if you first give everyone the placebo and then the second time give everyone the supplement with the active ingredient, and then it seems like everyone does better the second time with the active ingredient, but maybe it's just because people now better know how the test works, or because everyone the first time you did it at the end of the summer where everyone was in the off season, and then the second time they've trained another 3 weeks, for example, so everyone is just more trained. So by flip-flopping, like some people do it in one order, some people in the other order, any outside influence is just averaged out. Maybe there is some advantage from outside influence, but for one people that's an advantage, someone else it's a disadvantage, and that equals out. Placebo, what's that?
I think I already quickly mentioned it, but it basically means that besides the intervention, you have, or let's say next to your active ingredient that you want to study. So in this case, it's the nitrate from beetroot juice. You have another intervention which does not have the active ingredient. So in this case, they have beetroot juice completely depleted from nitrate. So they basically get the same drink, but it does not have the active ingredient.
So why is this better than just giving water?
So if you would give water, then it will be very clear what the intervention would be, or what the beetroot juice would be, and if you would get the water. And if you already explain the study and you explain that you think that beetroot juice will improve performance, well, then you can have the placebo effect of someone thinking, hey, this tastes pretty bad, so it must be the nitrate or the beetroot juice. So this must improve my performance. And then they can often already improve better or perform better just because they think that they're getting something that will improve performance. So that is the placebo effect, of course, and you want to control for that.
Okay.
And then, it's a crossover study, and in this case, it just means that the participants, they perform both of the trials. So they do all the testing when they get the beetroot juice with nitrate, but they also do all the testing when getting the placebo drink, which is the beetroot juice without nitrate.
So why not just 20 people with beetroot juice and 20 people without beetroot juice? Why twice on the same subject?
You could also do it just fully split up and not let all the participants do both trials. But by using the same participant twice, you minimize variation because you can kind of compare the same person on two occasions between the two drinks. If you would have a group with the one drink and a different group with the other drink, then you already can have some differences in the groups. I think it is possible, but then you would probably also need more participants. And in this case, it's very possible to let the same person do both of the trials. So I think it is a good choice in this case.
Yeah. So yeah, you already mentioned once the beetroot juice with and once without the active ingredient, and then they mention a washout period. So what's a washout period?
So in this case, for example, if you would have someone get the supplementation with the nitrate, so the beetroot juice with nitrate, then do all the testing, and then the day after they would do the placebo drink, so the beetroot juice without nitrate, and then do all the testing again. There is a possibility that the effect of the previous drink is still in effect during the second trial. So that is why you want a washout, so that you are sure that there's enough time in between that one of the treatments did not affect the other. And I think it kind of depends on the context, how long it should be. And I think here at least 8 days. I think, I'm actually not sure how they get to the 8 days, but probably at some point someone investigated it and said, okay, after 8 days, you probably see that the levels of nitrate in the body are back to baseline, and then you could say, okay, you are, let's say, back to baseline and ready for a next treatment.
Yeah, so it depends, like you say, it depends entirely on what you're researching and how long it stays in the body. If you test a supplement like creatine that stays much longer in the body, 8 days wouldn't be enough. If you study the effect of amino acid supplement, that stays in your body for a couple of hours, so you wouldn't even need 8 days, for example. Okay, next section. As you can see, I haven't really highlighted here anything. Here they just describe standardization of physical activity and diet. I think this kind of speaks for itself. If you want to do a study, you don't want one subject doing a 6-hour bodybuilding workout the day before and the other subject not, or in this case, since subjects do both experiments, you don't want them to do a 6-hour bodybuilding workout one day before one test and then do something and then don't do any exercise the second time before. Same with nutrition. You don't want them to eat tons of carbohydrates the days before one test and then the other test eat no carbohydrate whatsoever. You basically want them to do more or less the same. So subjects should do more or less the same, and then for both test subjects should more or less do the same. So this whole paragraph just describes which instructions the subjects were given. So for example, no alcohol. That seems pretty obvious. You don't want someone partying the night before a test and someone else getting a good night's sleep. One interesting one is, I haven't highlighted here, but where is it? So subjects were not allowed to use antibacterial mouthwash or toothpaste or chewing gum. And the reason is that the nitrate, to convert it into nitric oxide, so that pathway we discussed earlier, you actually need some bacteria in your mouth. So some bacteria in your mouth are bad because they result in caries, I think is the the official term. So they're just bad for your teeth, for your dental health. But some bacteria have good properties, and some of the bacteria help to activate nitrate into nitric oxide. So if you kill all the bacteria the evening before, then you can drink beetroot juice the next day, but it's not going to do as much because there's not enough bacteria to activate it. So just stuff like that, everything that possibly influenced the results should be kept the same as much as possible between the two tests and between subjects. So that's what's specified here. And here is the experimental protocol. So this just basically discussed what have we exactly done and at which time point. So have we taken blood samples? How much beetroot juice did the subjects got? What was the performance test? As you see here, so they say we did this yo-yo IR1 test as previously described. And that's perfectly fine. Not every paper has to describe in 3 pages how a performance test works, why it is legit. You can, of course, reference to other papers. But just as with the pathway, just reading about certain tests can be quite confusing from text. So it's often useful to, again, see it visually. So what is this yo-yo intermittent test? It's basically a repeated sprint test, and therefore it kind of resembles how a soccer player would act during a soccer match. So you start here at this start line, you'll hear a beep, and then you sprint to here, you immediately return, so you just have to touch or cross this line, immediately sprint back. Before you hear another beep, before that beep, you have to cross this line. So you sprint, instantly go back, and then you have to cross this line. And this part, once you hear this second beep and you finished in time, you have like 10 seconds to get ready for the next sprint. So usually here you're decelerating because you need to go all out to get over this line. You're decelerating, you're slowly walking back. Usually have like a couple seconds just to catch a deep breath, get into position, and then boom, you'll do it again. And then you do like 5 sprints at a certain speed, and then you finish that level. Then the next level, you do the exact same thing, except you just have less time to finish the sprint. So the further you get in the test, one, you become exhausted because you're doing more work, of course, but also you have to go faster and faster to keep going. So it's a combination of your sprint capacity, but there's also a little bit of an endurance component. And that's, of course, what soccer is as well. So that's what they're discussing here, how this test works, etc., how everything was standardized. Here they're discussing plasma and saliva analysis. So just to give a detail, the study needs to be reproducible, and basically every small detail needs to be known. So if you have blood samples, you, for example, centrifuge them to get the samples ready for analysis. Now you need to know exactly how fast do you centrifuge them, how long, what's the temperature. All those details are interesting. And then usually you see like some details are described and then a reference to a more comprehensive article that describes everything in more details, like the basis of what is this, how does that work. They're not going to explain that here. Then statistical analysis. I don't want to go too much into this because that's a topic on its own. But what's interesting is they say sample size of 40 subjects, including a 20% dropout was calculated. This is something, a lot of papers don't do this, but I think they should. What determines how many subjects you have in a study? Why does this study not have 200 subjects? And why does it not have 5 subjects? Well, a good study comes up with an educated guess how much subjects it needs and then uses that number, basically. Having 200 subjects, well, you can say that's better, but if you only need 40 subjects, you could have done 5 studies with those 200 subjects. So you kind of wasted a lot of time and money if you have unnecessary many subjects. However, if you just do 5 subjects, you might come to the wrong conclusion simply because you have too little subjects. So when is it too little and when is it too much? Well, that depends on how big the effect is of your intervention. So here they say we expected a 4.2% difference in the test based on previous data. And then again, I won't go into all the specifics, but based on this difference, you can calculate, now you need about 40 subjects to have a reasonable chance to demonstrate that indeed Beetroot is effective. Now, if Beetroot would increase performance by 200%, first of all, that's just completely unrealistic, but let's say that it improved performance with 200%. Then you don't need 40 subjects. It would probably with 5 subjects, you're like, wow, these 5 subjects crushed it when they had the beetroot juice. So it would be very simple to show that the supplement was effective. So the less strong the effect is of the supplement, the more subjects you need. But don't use 200 subjects when that's not necessary, because you could have done 5 studies. So you're just wasting time and money as well. Okay, on to the results. Milan, this might be as boring as methods, I think, or at least the text version of it. What do you think of the results?
Well, I mean, like you said, the text of the results is not usually the most exciting. I think if you read this, already the first sentence, I would be very relieved, because I already see that they are referencing to figures, and then I think, yes, let's go to the figures.
So here, I completely agree. So they mention something, you're like, yeah, sure, and then you click on the figure, and it's like, okay, it's pretty clear that Beetroot is doing something compared to that placebo. So visual, again, just kicks ass compared to text. But basically, we're going to be, as you see, I haven't really highlighted much. The function of our results is just to show your results. Again, don't say this increased something. You want to see specific numbers. You want to see specific statistics, so people know exactly what was the case. And ideally, you also show your data with a lot of figures, because just much better to get a grip of the data. So looking at this figure 1, Milan, can you explain why they measured these things and basically what the result is and whether or not we should care about that?
So here you see the metabolite concentration, so nitrate and nitrite, both in the plasma and in the saliva. So I believe they measured this 2.5 hours after the ingestion of the final supplement. So I would say this is basically just a check whether the supplement indeed is working, or at least showing something in the body that should happen. So if you give the beetroot juice without nitrate, then plasma concentration should be relatively low, and when you give the beetroot juice with nitrate, then the concentration should be high. If these bars would be the same, then you would have a problem, because even if you would show an effect, then where's the effect coming from? You basically have no clue. Yeah. So I think this is just the basics, showing that there is indeed something happening in the body that we expected. So this just shows us that.
So if we go back here, so basically the hypothesis is you ingest nitrate, that gets converted to nitrite, that gets converted to nitric oxide, and that then can have performance enhancing effects. So nitric oxide is very difficult to measure, because it only exists for a microsecond, and then it's converted into metabolites, so you can't really measure it. These are quite easy to measure. But if you take a supplement that has nitrate and you don't see an increase in plasma nitrate, then you have to start wondering what's happening. Maybe the supplement didn't actually contain nitrate, maybe a production error happened. So basically you want to see every step of the way, so you get just a much better insight of what's happening. So here it's pretty clear, like the supplement does what it's supposed to do. The supplement has nitrate, and indeed you see that nitrate goes up both in the plasma and in the saliva. Why is saliva interesting, Milan? Do you know?
I mean, usually measuring something in saliva is easier. So you just have the cotton swabs that you put in the mouth, and then yeah, they collect saliva and they can measure stuff, which is basically easier to do. And even a participant or an athlete could do that themselves. If you measure plasma, you need to take blood, you need to centrifuge it to get the plasma, you need to freeze it, you need to analyze it, but it just takes more time and I think more money. Saliva is more convenient usually, and I think in this case they also measured it to compare the two, to see whether the values that you get from saliva are kind of in line with the values from plasma.
Yeah, but there's a few other reasons. So one, plasma is more invasive, like spitting into something is pretty easy. A blood draw is a bit more invasive. Like I know for us, it's like a joke, that's nothing, but there's plenty of people who are afraid of needles. And if you work like in some populations, so for example, the higher the level of the athletes is, by definition, the less of that population is available. And then if also some of them are, for example, afraid or not very keen on a blood draw, you lose more and more subject numbers. So sometimes you just take saliva because you can't get enough plasma samples. But that's also not the main reason. The main reason in this study why they are interested in saliva is before we mention that your oral bacteria have to activate the nitrate. So that's why saliva concentration is interesting, because your mouth is where a lot of the magic is happening. So in this case, saliva is a pretty interesting measurement. Because the oral cavity is just a very relevant metabolic site for this type of research. Okay, then the next result, probably the most interesting, well, not probably, the most important one. What do we see here, Milan?
So here we have the yo-yo test, which you already explained. So this should tell us something about soccer performance, basically. So if you look at panel A, you see the placebo or the beetroot juice. And you see that the black bar, so the beetroot juice, is a bit higher, and it has like the nice asterisk on top of it, which usually indicates that it is significantly higher. So with all the statistics, it showed that there indeed seems to be a positive effect from beetroot juice. I think here it is good to have a quick look at the y-axis, because there is like this small gap. Because if you just look at the bars, it seems that there's a relatively big improvement from beetroot juice. But this, if you look at the text as well, they also specified it's actually 3.4%, even though it kind of looks more like maybe 30%. But they just did it so you can visualize the difference, because the performance is based on meters, and it's quite high numbers, so it's around 1600. If you would have this y-axis from 0 to 1700, then it would just be like two basically almost identical bars. So this is just to kind of visualize the difference.
Yeah. So I think that's it's good to mention. So always look at this axis, what it represents, and definitely don't make the mistake. Oh, it's 33% higher. what I also think is very important with any graph is that you look at what are they measuring, what is the unit, and what does that actually represent. So here you see that the beetroot juice has a higher number. Is that what you would expect? So if we go back to that yo-yo test where there were repeated sprints, and you basically have to get over a line before the beep can be heard. If you fail to do that, you're done. So if you do more distance, it just means you were able to do more of those sprints in total. So in a test like that, a higher distance means you're able to keep up your sprint levels longer than with the placebo. So a higher number is good. In contrast, there's plenty of performance tests where you want something to be lower, and then, for example, the time to complete 1 mile, there you want a bar to be lower. So you always have to think very quickly, like, okay, you see a difference. Is that difference a good thing or a bad thing? Here, it's a good thing because it means you are able to do more sprints in total. Second one, Milan, what's B?
So B basically shows the same data, but it now shows all the individual participants. So on the left, you see then the participant doing the test with the placebo trial, and then they are actually connected to themselves. On the right, when they did the beetroot juice trial. In this case, I think they also kind of made it easier to visualize what is happening by having different lines. So they have like the solid lines, all the participants showing a better performance with the beetroot juice. And then with the dotted lines, it's more similar results. I think this
so it's either similar or worse.
Yeah, exactly. So I think this is really nice, and I do really like it because it gives a bit more information about the data.
Okay, so what is the main information that you get here that you wouldn't necessarily get here?
Well, if you look on the left, you just see the mean with the standard error of the mean. But it could be that maybe a couple participants are showing huge improvements, and the rest of the participants are kind of similar, and then you could get the same like dynamite plot that you see on the left. But if you show like all the individual results, then you do see that, okay, most of the participants seem to be like slightly improving their performance, even though a couple of participants are showing similar or sometimes even a bit worse performance.
We'll discuss the results more at the discussion section, but I think we kind of discussed how to look at this graph. Gastrointestinal and Borg scale. So these are just questionnaires about the Borg scale is how intense do you experience the exercise, and the GI is just is your stomach upset because beetroot juice is quite the specific taste. Most people don't find it very enjoyable. I have some beetroot here that I'll have a glass. I've had plenty of beetroot. This is just something random I got in the supermarket for this paper. We used different beetroots, which I'll show you later. I just want to relive the experience again. I also participated in a study with beetroot. I think you as well, Milan, or not?
No, not
beetroot. So just to briefly address that, we did a study where we looked at different vegetables that were rich in beetroot. They were all gross, all drinks, but beetroot was the least gross. But it was a lot of fun because there's just a lot of people at the same time in the lab, and people were not too happy about the drinks. But like it's just important to know because a supplement can improve performance, but if it improves performance by 0.2%, for example, but everyone hates it, you probably know it's not really going to do much in practice because people are not going to take it anyway. This correlation analysis is not super interesting. They were just looking whether or not the plasma and saliva gave the same result, but that didn't really seem to happen. So some subjects were really high in plasma, some were had very high levels in metabolites, but they were not necessarily the same subject. So some subjects were high in one, but low in the other. Why that is, we'll get into in the discussion. So discussion, Milan, what's the purpose of it?
So with the discussion, you can kind of show the results in that context. So with the results, you just give the results and nothing more. With the discussion, you can kind of cheers. But with the discussion, you can kind of discuss what the results mean. So if you show certain effects happening, you can really kind of like tell a story about it, that what you think it means, and also kind of compare it to other studies that have been done, and if it was what you expected, or were there maybe things that you didn't expect, basically everything around that.
Okay, so structure of a discussion, it's not like an introduction that it is a funnel shape, for example. First paragraph, what does the first paragraph usually describe?
So yeah, you can see it here, and it is usually the case that the first paragraph just gives the main result, so the main finding of the study. So here you can also see that the current study demonstrates this and this and this. So it's just the main finding right from the start, and then the second paragraph. So after that, you can see that they also give I think this is more like a short summary from the introduction, to just give that background again very shortly, just to kind of remind the reader why you investigated this, and also just a brief summary of the introduction before you go into all the results. And also as you can see here, they just have a paragraph for all the results, and also discuss it, whether it's you can discuss stuff, whether it's what you expected. You can also discuss it in terms of whether it compares to other studies. And I think you're pretty much free to discuss what you think is relevant.
Yeah, so in general, like you mentioned, you start with a result, and that gets its own paragraph. So here, the result was that the beetroot supplementation increased the relevant metabolites. And then they discuss, well, the increase was this big, and that is in line with previous research. So that's often what happens. This was our result. Was that in line with different research? Well, in this case, it is in line. If it's not in line, you would have to try to explain why. So you would say like, oh, we gave a lower dose than those studies, or well, our study was in trained subjects, so maybe our trained subjects metabolize these things faster and therefore it's lower. So your result, compare it to the literature, and then if necessary give some kind of explanation of why it's the same or different. Just as with the results, they use the same order. So before we're even gonna look whether or not there was a performance enhancing effect, let's first see if the step before that the metabolites act the way they should, and in this case they did. So then here they mention that metabolites went up, great, but what we're really interested was that we saw a quantifiable improvement in high intensity intermittent running performance. So the supplement does what it did. Again, they mention the specific number and then they compare whether or not that effect and number is in line with previous studies. So the exact same trick they did in the previous paragraph, they do it now, but just for a different outcome. And then everything else is just explanation of why this number makes sense or not makes sense. So here they discuss a little bit of that theory again, that because it's high intensity exercise, they expect that this number is relatively high, that for endurance exercise it might be a little bit lower, in trained subjects it might be a little bit smaller than in untrained subjects, but not that much, etc., etc. Now, to be honest, I would wish that somewhere here they ended that paragraph and then just started a new paragraph for this next point where they say, clearly, such a performance benefit should be attained without any major negative side effects. So in my mind, that's kind of like a different point. So I think it would be more clear if this was a different, paragraph. But of course, it's an important point to raise. Like, okay, our supplement seems to have positive effects, but are there negative effects? So then they discuss their data on GI discomfort, etc., etc. Speaking of which, I have to say this beetroot juice is pretty decent. That's probably because it's very unconcentrated, which is something I'll get back to you with in a second. Here they mention, we also mentioned heart rate. I don't think that's particularly interesting, so I'll leave it for now. For the journal club, the main important thing is that they do the same thing. So we see something on heart rate, how does that compare to other studies? And then what is different about our study than those other studies that might explain it? Here they mention why nitrate supplementation increases plasma and saliva nitrate, but that these two things don't seem to correlate very well. And then they explain why they think that is. The reason is because they just collected one blood sample prior to the exercise test, and it's possible, for example, that the values first go up in blood and then in saliva, or vice versa. So maybe the peak values in blood and saliva would correlate very well, but you would have to have a timeline to see that, which they couldn't do in this study, because of course you can't take blood samples while your subjects are doing sprint tests. So they say like both things are fine to check if the metabolites go up, to confirm that part, but just don't necessarily translate the value from one to the other. Maybe that can happen, but for that we need more information and a better timeline. Then finally, towards the end, there's usually a paragraph, one or two paragraphs that discuss more like practical implications of the study. So before it was, this were our results and this compares like this with other research. And then towards the end, often it's discussed what the practical implications is. So here they say we use this yo-yo performance test because it has been shown that that strongly correlates with the ability to sprint during a soccer match. So a much easier test is to just put subjects on a bike and just let them cycle until they can't cycle no more at a certain resistance. It's much easier to do, but that's not how soccer works. So you always want to do a test that resembles the real sport as much as possible. One limitation here is that actual soccer performance is literally, are we winning matches and or how many goals are we making? Like that's actual soccer performance. Well, you can never ever do any, scientific research on that, because even if you would say, well, one season we're going to take beetroot juice and the other season we're not, do you have the same soccer players? Are they just as fit this year? Your opponents are not the same teams. Your opponents are doing all kinds of different things. So you can never have a controlled environment in an actual soccer match. So the best thing you can do is have a controlled environment in an exercise performance test that resembles soccer. But you can't say, well, performance went up by 3.4%, so you're going to win 3.4% more matches. You can't say that. Clearly, if you're better at repeated sprints, that has the potential to translate into more soccer wins. Theoretically, it could actually translate in 50% more soccer wins, because if that 3.4% helps you to be slightly faster than your opponent, and you just beat them consistently, that might be a lot of wins. Like, you only need to be a little bit faster than your opponent, and that can completely change the whole match. But the main message here is that you can just never say, oh, this trends like that into more competition wins. Here they also say that previous research suggests that Beetroot was pretty effective in untrained subjects. We now showed that it was trained in pretty decently trained subjects, but of course we still haven't investigated elite level athletes, so that could be a next step. Now, why is this so interesting? Because research has been done, quite a lot of research has been done on endurance exercise, and for endurance exercise it's pretty clear that beetroot juice also improves performance, but only in relatively untrained subjects. So you see that in trained and especially in elite level endurance athletes, beetroot juice doesn't do a whole lot. So our study in soccer shows that with intermittent type exercise, training status doesn't seem to be that important of a factor, but in endurance exercise it does. So that's why we're saying, well, training status might not be that important for this, but we still should confirm whether that's also true for the elite level soccer players. Milan, are there some, let's say that you just had unlimited money and time, what are some things you would have done different in this study? Like, this is always the easiest thing. Making studies better is not that difficult if you just have more time, money, subjects are okay with it. But just for fun, as an exercise, let's brainstorm a few things that would make the study better, not considering resources.
Well, what I always like to see in a study is when they take muscle biopsies.
Yeah, so we're in muscle metabolism. So just as that simple check with the saliva, we also like to see, okay, if the muscle is supposed to perform better, what happens to the metabolites in the muscle, etc., etc. So yeah, I 100% agree. Of course, you would love to see that. I can quickly address why that's not possible here. One, that would make so the muscle biopsy kind of messes with the performance. Don't get me wrong, a muscle biopsy hardly it sounds very invasive. It's hardly invasive, but if performance is your main outcome, you kind of should stick with one main outcome. So either you're interested in performance and then don't do anything that can mess with that, even if it's just in the head of your subject, or you're interested in the metabolic effect and then focus on that and then just leave the exercise test for another study. So you kind of have to choose there. The other reason is again, a muscle biopsy is not that invasive, but you do have to make sure that it's in a hygienic environment, etc. And this study was not performed at the university. So after working hours, we went to the actual soccer clubs, so that the performance tests were performed in at their own clubs where they normally perform. So everything was very familiar for all the subjects. And in an environment like that, it's just not suited for muscle biopsies. You have to do that in a controlled university setting, of course. So yeah, that's one thing. Anything else you would love to see?
I mean, I wouldn't say more participants, because I think they still show an effect. So I don't think
So
it's
why is that
necessary.
important? So if they wouldn't have shown an effect, would more subjects be interesting or?
Yeah, so quite often if you see a study and they don't see an effect and you look at the design, you often see that it's a relatively low amount of subjects, and that can be an issue because if you have a low amount of subjects, then the chance is just um, higher that you will not find or detect the effect that you're looking for. So this is like a statistical issue. If you have low number of
Oh
subjects,
boy, the viewers are dropping.
variation. No, that was just my point.
Yeah,
Like
no,
if you have
it's a very good
low
point.
amount of subject.
It's a very good point. So we're not gonna bore everyone with the statistics, but the basic principle is that you need convincing data whether something is improving something. Otherwise, the default conclusion is no, nothing is happening. So if you do a study with just a handful of subjects, by definition, you don't have convincing data because you only had a handful of subjects. But like Milan said, in this study, there was a significant effect. So by definition, they didn't have too little subjects. Still, more subjects, it's always better. But here, the added effect of more subjects, I don't think it would have added a whole lot more. One thing that you rarely see this because it costs way too much time and money, but one thing that theoretically would be super cool is subjects did one test with the placebo and then did one test with Beetroot. So, for example, if I look at this subject, it seems like this subject did worse with the Beetroot. Then what you sometimes hear is that people say, well, you have either no responders or people who even respond negatively to the supplement. Well, that's unlikely what happened. I wouldn't know of why someone, like I wouldn't know what the metabolic effect of beetroot juice is, why someone would possibly perform worse. The only thing that I could remotely think of is that they got sick of the got sick of the drink. So there's just no good reason to think why anyone would perform worse. It's most likely that this person just had a bad day because not everyone performs exactly the same each day. And maybe without the beetroot juice, he would have performed even worse. So maybe it would have been like this if he didn't have the beetroot juice. So one thing that theoretically is super cool, but you rarely see because it just takes too much time of the researchers, the subjects, and too much money, is to do 2 placebo tests for each subject and then 2 beetroot juice tests. Because if you see like 2 dots very close next to each other here and 2 very close here, then you're pretty certain, like it's very clear that this subject consistently performs better with beetroot juice. And if you would do that, my guess would be that this subject would have 2 dots pretty close to each other with the placebo, and then here he had his bad day, but then he would also have a good day, and then on average you still see that also for this subject beetroot juice actually is useful. So that's something really cool, but you can do that. You can simply do more subjects. There's tons of options, but it's interesting to think of. Okay, I think we discussed most of the study. Maybe it's interesting to discuss a little bit about conflict of interest. Any comments, Milan?
Yeah, I mean, you very often see on, especially on social media, that if there is a conflict of interest, that people somehow think that the whole study has no value, that it was for sure influenced by, yeah, other parties, and that it somehow affected the results and stuff. And I think that is really just not the case. I think how it usually goes is that a research group just does a study with a company, and that the company usually has something to say about the design, but how the study is performed, how the data is collected, and everything around that is usually in the hands of the research group. And yeah, I think that sometimes it does tell you to be a bit more skeptical of the results, but I don't think it's really a reason to say that the results are false or whatever, what you quite often see online.
Yeah, so again, it's just an indicator like, okay, there was some bias. I think you can say that. Clearly, the company wants a good result. So they would be interested in designing a study where a good result is more likely. But that doesn't mean the way the study was done is wrong or anything. It's just, okay, under these circumstances, a positive effect can be found, for example. That effect, that is likely very true. As a reader, you should just think, what they exactly tested, is that interesting, is that relevant for real life? So what you, for example, often see is that, let's say a supplement company sells a certain mineral, and then they do a study where they give that mineral to subjects that were highly deficient in that mineral, and then they say, see, this improves performance or some hormone in the body. And while that study is legit, it just doesn't show that everyone should supplement with that mineral. It only shows that if you are deficient, then yes, the supplement could be interesting. So always just critically look at the study, but don't dismiss it because of funding. So in this case, it was funded by the government, so I don't think there's any concerns about commercial interest. We have a comment that perhaps a longer study would be interesting. So a longer, like there's a few ways to look at that. So in this study, they did 6 days of supplementation. So you can see, what if you supplement for 12 days, would you maybe not a 3.4, but maybe a 5 or a 6.8% increase. So that's one thing. To keep it short, it doesn't really seem that if you supplement longer that the acute performance increases. However, what you can also do is a longer study, as in several weeks or months, and see, okay, beetroot juice, just taking it for a few days can improve performance. But what if you take it before every training? Do you get a little bit more out of training, and therefore that effect builds up over time? So if I can sprint a little bit better, does my sprinting capacity improve over time? Because every sprint training, I'm a little bit better than someone else who isn't taking beetroot juice. So in that sense, a long-term study would be very interesting. But it's a little bit of a different question. So this study just shows that if you take beetroot, it gets metabolized, and basically there's an instant performance enhancing effect. And then another question would be, so this study suggests you want to take it, the couple days before an important match. Then another question is, if I consistently take this before training, do I get more out of my training and become better over time? Because you can also speculate, maybe your body gets used to it and the performance enhancing effect goes away, and then you definitely do not want to take it every time. But I wouldn't say that's not necessarily a better study. It's a slightly different study, and you would need to do both types of studies to get a complete picture. Maybe one more thing I want to show. So here you see the beetroot shots that were used in this study. I don't have any at home right now, but this is a pretty small bottle. It's like, so here's my glass. That bottle will be like this big and pretty thin. It's like, really, it's a shot. It's a beetroot, it's also on here, concentrated beetroot shot. And that little shot would probably have just as much nitrate as this entire bottle. So it's really made as a practical supplement to get enough of this nitrate. So we used this, and then we asked the producer of this to make the same supplement where the nitrate was removed from this bottle. So we basically had identical bottles. One's the beetroot shot with nitrate and once without. Now, I quickly want to address some practical recommendations. We've really done like a journal club now, like what should you look at in a paper, how should you read it. Just want to quickly do one slide with some practical recommendations. Maybe you're convinced that beetroot juice could be relevant for you. So these recommendations are not based on the paper we just discussed. This is based on a paper by Jones in Medicine, Science and Sport Exercise. That's the journal, and it's a pretty recent review. And this author is the person who has done the most amount of research on beetroot juice, and also one of our colleagues is on this paper. So that's where I'm basing my recommendations on. So basically I'm just telling their recommendations. So they suggest that the dose of beetroot juice should be at least 300 milligrams of nitrate. Here you see that one of these shots has 400. In studies, often one or two of these shots are used. So I would probably take 400 as minimum, and I take one or two of these shots. So keep in mind that not all beetroot juice is the same. So if you take something as this, this product doesn't mention how much nitrate is in there. It does mention like this is this much juice, which translates to this many grams of beetroot. So you could kind of calculate it, but I don't think there are many people who use unconcentrated beetroot juice because you just need quite a lot and it's not really convenient. So in practice, people always use 1 to 2 of these shots. Do you need to load? So loading just means, do you need to take it for several days, or can you just take it on your exercise day? Most studies, like the study we just discussed, use a 6-day protocol, but several studies have shown that one dose just before exercise can already be effective. So if you want to play it safe, just take it the 6 days before your competition, but one dose might already be all you need. Then the timing, they say if you use beetroot juice, take it at least 90 minutes prior to exercise, because this gives enough time where the nitrate goes through your blood, through your saliva, the bacteria can already convert it to nitrate, so it's basically ready to be converted into that next step, nitric oxide, when you do your actual exercise. If you consume vegetables, so let's say you eat a lot of either beetroot or spinach, then the digestion needs a little bit more time, so then they recommend 3 hours prior to exercise. Again, I'm not very sure if many people would do this, because you would have to eat quite a lot of vegetables and it would be like a major hassle and distraction from your competition, but at least theoretically you could do it, but you have to eat it a little bit earlier. And then finally, who can benefit from this? So untrained or moderately trained endurance athletes. So I discussed earlier that elite endurance athletes don't really benefit from beetroot juice, likely because their bodies already have optimal nitric oxide amounts just as a result of their training. Moderately trained endurance athletes benefit a little bit, not as much as untrained, but you still see a little bit of a benefit. But elite athletes, you see no benefit. Now with intermittent type exercise, it's a little bit different because here it seems that even high level athletes can see some benefit. So since the study that we discussed, some other studies have been done, and it seems that during high intensity exercise, basically anyone can benefit from it. And as a final note, for all supplements that you use, check the doping list and/or contamination. Either if you're an athlete that competes or you're a coach and you recommend athletes, make sure that one, your supplement is not on doping list, which is not the case for beetroot juice or nitrate, but also be aware of contamination. So here you see Informed Sport, which is a website that records for a lot of supplements say, okay, we tested this supplement, this batch number produced at this date, and there were no illegal substances in the supplement, because it's scary how high the percentage is of supplements that have illegal compounds in it, and that can ruin your career. So always check this site if you are interested in using any kind of supplement. I think that was all that I had to say. Milan, do you have any other comments?
No, I think you had a nice overview of the practical recommendations, and I think it would be interesting to see this kind of study performed in like the elite athletes. I don't think it has been done that much.
Not for
Big
soccer,
football.
but it has not done for soccer, but they have done it in BMX cycling, which is basically like sprint cycling. It's not like Tour de France cycling, and there it can have certain benefits. I don't want to go into too much detail. But yeah, some elite athletes seem to respond to it. Let's keep it like that. So you cannot conclude like it only works for noobs. No, but it seems to be depending on what type of training you're doing and whether or not your body is used to having a lot of nitric oxide.
Hmm. Oh yeah. No, I don't have anything to add.
Perfect. So we basically finished 2 hours. If there's no other questions from chat, and then I think we're gonna wrap it up, and we're gonna pick a new paper. Let people know what we're doing next time. If they just go watch this video, we'll upload this video to YouTube so you can watch it in your own time. And most important thing is just go through which steps did we take. If you do that for another paper, I'm pretty convinced that you should be able to figure most things out. So, a question is coming. Someone says they have a question, so let's just wait. So Milan, tell a story in the meantime.
Oh. Mm. So I was in a hotel the other day, and we had like a mountain biking package. So I was expecting like a big pasta meal, and then in the evening we got like this tiny lasagna, and that was it. And you had like a whole day of cycling, and then you get like the one tiny thing, and I really thought like this has to be like the appetizer, but it was the whole meal.
But Milan, I've had several meals with you, and anytime I've served you dinner, you're like, "Is this it?" So I think it's more your issue than the hotel. Okay, so the question we get is: compared to other supplements like caffeine, what would you recommend more, or would you take both? Um, well, caffeine is so the number one supplement that has been researched the most is creatine, and that's mostly interesting for strength sports. Uh, Other than that, caffeine is the most researched supplements, and I mean for decades, while all beetroot juice is from the last decade. So caffeine just has a lot more evidence that it can be performance enhancing under a lot of conditions. So if I would have to pick, I would pick caffeine. However, I think these supplements, at least theoretically, can work synergistically. There's one study who looked at it. It didn't find any synergistic effect, but most likely, that's my guess, is because they used very trained subjects in endurance exercise where beetroot doesn't seem that effective. But caffeine mostly works by suppressing the feeling of fatigue in your brain. So it basically just blocks the feeling of fatigue, while beetroot juice actually does something in muscle. It allows through the nitric oxide. It allows more blood flow to the muscle. Also seems that you can use oxygen a little bit more effective in muscle, and it also improves the contractibility of muscle tissue. So there's several effects that are thought to mediate the effect of nitric oxide on muscle. But since one works in the brain and one works in muscle, I don't see a reason why these two cannot together have a better effect than each one separately. But yeah, it's just difficult because if you want to do studies like that, you kind of need 4 groups. So you need a control group, then you need one group with nitrate only, then you need one group with caffeine only, and a combination of both. And then you need so many subjects that it's just very difficult to have enough subjects to really figure out. And because most supplements just have a relatively small effect, like what we saw here, 3.4%, it's just very difficult to do these types of studies. So in practice, yes, I would recommend both. There's just no specific study saying that it would work. So it's anyone's guess at this point. Okay. I think we're going to wrap it up. Everyone, thanks for watching. And hopefully we'll see you next month.